Good day everyone, welcome back to my channel. Today we shall discuss a small yet really important topic from the chapter Principles of Inheritance and Variation. This topic is very important catering to your board examination as well as NEET preparation. So this topic is incomplete dominance. You may ask what incomplete dominance is. So in one line we can say that it is a phenomenon where F1 hybrid, this will be F1 hybrid, F1 hybrid exhibits characters intermediate of the parental generation. Now I know this is quite bookish definition, and in terms of explaining, it's basically where the F1 generation or the first progeny, it basically has the characters or the traits that resembles none of the parental traits. It's basically an intermediate or a mixture. It seems to be a mixture, though I'll tell you why it's not mixture and it's not blending. So, and then where we see them, you will find this in plants like Mirabilis jalapa, which is also known as 4 o'clock plant, and Antirrhinum majus, which is snapdragon. Now, why we are calling this thing to be incomplete dominance and what exactly we see in these plants? So let me explain this to you. In these plants, red color flower is dominant and its genetic constitution is capital R, and white is recessive and having the genetic constitution smaller. Now you know that in general cases, in Mendel's hybridization experiment of the pea plant, we saw that the F1 generation, I'm writing this in Mendel's pea plant experiment, the F1 generation had capital T small t and the phenotype was tall. So basically it had the dominant trait. Now you may assume that the case will be similar in case of this, that the F1 generation is capital R smaller, as we know. This is parent, F1 is capital R smaller. Now here comes the twist. This color will be pink, and as you know that pink is neither red nor white, it's a mixture of red and white, so it resembles neither of the parental trait. Now comes another thing, that is there blending occurring? Like this thing already is violating the law of dominance that Mendel provided with his experiments. Now I'll tell you that there is no blending here. Now why there is no blending? So let us cross two plants, two pink color plants like we did in F1. So we have this capital R smaller and capital smaller again. We will get capital R, capital R smaller, capital R smaller, and smaller. So smaller is white, capital R is red, and this two, capital R smaller, is pink. Just a second. This will be pink. So As you can see, in F2 generation, we are getting back the parental traits. So that's why we can conclude that there is no blending, and the law of not being blending is universal. So we are getting the phenotypic ratio as 1:2:1, which is red, pink and white. And surprisingly, genotypic ratio is also the same, which is capital R, capital R smaller and smaller. So writing this in a neat way, I'll write this in a neat way. Parents is, we have red flower and white flower. Red flower is capital R, white flower is smaller. When we cross them, we get the gametes as capital R and smaller, and we get capital R smaller in F1, and all are pink. And when we do selfing of them, I'm showing the selfing in the next page. In the form of a Punnett square. Pink, capital R smaller is again pink, and smaller is white. Phenotypic ratio, genotypic ratio, both are 1:2:1. Again 1:2:1. So now I told you that this topic is important for boards and NEET, and in what sense they are important. What you need to remember, first of all, remember the examples. That is, I told that it's Mirabilis jalapa, 4 o'clock plant, and Antirrhinum major snapdragon. Remember these examples. Secondly, remember the phenotypic ratio and also the genotypic ratio, because in NEET they may ask you questions like in what case the phenotypic and genotypic ratio is same, and then they can ask that they're giving this snapdragon or Mirabilis jalapa as one example. So by this video you can understand the significance of this topic, and I hope you like this video. Thank you.